Hello everyone, welcome back to my channel. I hope you all are doing well and you are preparing pretty well for the DBT-JRF examination. So this is the day 20 of the free BET crash course in which I am going to discuss some of the important questions from the genetics portion as per your request. I have also uploaded one video regarding the important topics from the genetics portion. So I will provide the link of that particular video in this video I button or the description box. So you can check that video also, and please hit the like button if you like my efforts, and you can also subscribe the channel if you want more from this. So let's start with the very first question, that is the segregation of alleles on one trait did not have any effect on the segregation of alleles on a different trait. This is based on. So basically in the question it is asked that the segregation of one allele trait did not have any effect or influence on the segregation of the alleles of a different trait. So what is this? Which law is representing by the given statement? So is it a law of heredity, or the law of dominance, or law of independent assortment, or law of segregation? So if you are aware with the Mendel law of independent assortment, you must know that this is the law of independent assortment by Mendel. So the correct answer is option C. It means the principle of independent assortment states that one allele at different loci separate independently of one another. It means the segregation of one allele is independent of the second allele. And this is what the law of independent assortment by Mendel. So correct option is option C, that is Mendel's law of independent assortment. Now question 2, that is genes that show tendency to be inherited together is known as. So the genes that show tendency to inherit together is known as the linkage group. So the correct answer is option A, and the genes which are packed together on a chromosome and tend to pass from one generation to another generation are known as the linked genes, and the genes are on the same chromosome and they passes on from one generation to the another generation, and they are known as the linkage group. So the correct answer for the question is linkage group, that is option A. Now question number 3. The giant polytene chromosome are found in, is it egg of fruit fly, salivary gland of larva of fruit fly, salivary gland of adult fruit fly, or all of the mentioned? So the giant polytene chromosome are exclusively found in the salivary gland of larva of a fruit fly, that is Drosophila. So let's see what exactly is this giant polytene chromosome. So basically polytene chromosomes are giant chromosomes which are found in certain tissues of Drosophila and some other organisms. You can also see here in this picture 11.8 polytene chromosome, which is a giant chromosome isolated from the salivary glands of larval Drosophila. You can see here the giant chromosome where the each puff separates a region of relaxed chromatin where transcription is taking place. This large unusual chromosomes arise when repeated rounds of DNA replication takes place without accompanying cell division. It means continuously DNA replication होता रहता है and cell division नहीं होता है वहाँ पे, बस DNA ही replicate होता जाता है. So it creates a puff and then producing thousands of copies of DNA that lie side by side. When polytene chromosomes are stained with dyes, numerous bands are revealed. Because so many DNA will occur after replication, so numerous bands हमें दिखते हैं किसी भी डाई से हम स्ट्रेन करते हैं तो, and it exhibits the structure known as the chromosomal puff. Due to the overload of DNA, it is represented as the chromosomal puff, which is a localized swelling of the chromosome, because of the repeated DNA replication. So polytene chromosome क्या है? एक giant chromosome है which is found in the larval stage of the Drosophila exclusively in the salivary gland. Now question number 4, which states that loops in Lampbrush chromosomes represent site of. So basically Lampbrush chromosomes जो होते हैं, Lampbrush chromosome occur like this. It has a structure like this. Folding like structure. Now you have to mention that the loops in the Lampbrush chromosome is a site for what? Replication, transcription, cell division or crossing over. So basically these Lampbrush chromosomes are a type of giant chromosome found in the growing oocyte of amphibians. So you have to remember the occurrence also. Now the twin loop arise, as I have already mentioned the structure. So this twin loop like structure on either side of the chromosome is for the active transcription of many genes. It means the Lampbrush chromosomes are transcriptionally active chromosomes found in the germinal vesicle of large oocytes of many vertebrate as well as invertebrate animals, and also in the giant single-celled alga, that is Acetabularia. So you have to remember the occurrence as well as the function and the structure. So the correct answer is answer B. It means the loop represents the active transcription site. Now question 5, that is, how can it be determined whether the parent progeny is homozygous or heterozygous? So this is an important question regarding the test cross. It means कि ये जो क्वेश्चन पूछा है वो ये पूछा है कि how you will determine that the parent progeny, पेरेंट का आप जीनोटाइप कैसे पता करेंगे कि इज इट हेट्रोजाइगस और होमोजाइगस. सो इट इज़ ओनली पॉसिबल थ्रू दी टेस्ट क्रॉस. इट मीन्स करेक्ट आंसर क्या है इसका? टेस्ट क्रॉस. So test cross is a cross of F1 generation offspring with the homozygous recessive parent. It means, suppose you are performing a cross between homozygous tall and homozygous dwarf. So you will get capital T and small t in the F1 generation. Then in the F2, if you will cross this capital T and small t with the recessive parent, that is small t and small t, then this type of cross is known as a test cross. It means when the F1 parent is crossed with the homozygous recessive parent, then it is known as the test cross. Test cross is the cross of F1 generation offspring with the homozygous recessive parent. If the test cross ratio is 1:1, it means the offspring phenotype is dominant is to recessive is 1:1, then it means कि जो parents हैं वो heterozygous dominant है. And if the plant exhibits dominant phenotype, the F1 offspring is homozygous dominant. This is how we can determine the genotype of the offspring from the test cross. Now let me explain this in a very easy manner. Suppose you have given a tall plant and you have no information that its parent is heterozygous. Its parent is either heterozygous or homozygous. आपको एक tall plant दे दिया गया है and you have said that this is a tall pea plant and you don't have any information about its parent genotype कि parent का genotype hetero था कि homo था. So what you will do is you will perform the test cross. Now you have given a tall plant and you don't have any idea about the plant is heterotall or homotall. So you will perform test cross to know the genotype. So the plant were homozygous, that is capital T, capital T. If through test cross you will get all the tall progenies, यानी कि उसके पेरेंट्स जो हैं वो होमोजाइगस टॉल होंगे अगर जो प्रोजेनीज हैं वो सभी टॉल मिले हैं. इट मीन्स कैपिटल T into then you will get this. and अगर जो टॉल प्लांट है वो हेट्रोजाइगस होगा जब हमें हाफ प्रोजेनीज टॉल मिलेंगी एंड हाफ प्रोजेनीज डॉर्फ मिलेंगी, यानी 50% टॉल एंड 50% डॉर्फ. तो इससे क्या पता चलेगा? इससे पता चलेगा कि जो पेरेंट थे वो हेट्रोजाइगस थे. इट मीन्स कैपिटल T एंड स्मॉल t. तो आई होप आज का वीडियो आपको अच्छा लगा हो एंड हिट द लाइक बटन अगर आपको ये वीडियो अच्छा लगा हो. एंड really very sorry for this short video because I am traveling and so you can understand. So please cooperate and hit the subscribe button if you want more from this channel. Thank you so much for watching.